Welcome back to Intro to Physical Anthropology. I'm your instructor, David Leitner, and today we are going to be talking about meiosis, which is the way that new sex cells are formed. We'll touch a little bit on chromosomes themselves, what they are, and some important aspects of them, and very briefly about mitosis, which is the way that somatic cells grow and divide. But mostly I want to sort of emphasize meiosis today because there are certain things about the process of meiosis that are directly relevant to the patterns of inheritance of genes as well as patterns of evolution. Finally, I will end up by talking very briefly about a couple of techniques that all of these discoveries in molecular genetics have enabled us to use to better understand the ancient past. In many ways, it has, over the past 25, 30 years, completely revolutionized physical anthropology. Even 10 years ago, the stuff I had learned 10 years before as an undergraduate was completely out of date. Like, that's how fast things have changed because of these new technologies. So we're going to be talking about it a lot. That said, let's get started. Okay. A few key terms here. Feel free to pause if you want to write this down. I'm going to be using several of these terms a lot. So, chromatin state, or chromatin. DNA doesn't spend all of its time coiled up the way we've been talking about, okay? Actually, most of the time, it's kind of a free-form, wibbly-wobbly sort of mess, just kind of hanging out in the nucleus, okay? It's only during the time when the cell is getting ready to reproduce itself and split apart that it coils up into chromosomes that we're familiar with, okay? Chromosomes are, the DNA doesn't sort of line up in a single long line. There's actually several separate strands, and the chromosomes are, those strands of DNA. Genes are always located on the same spot on a chromosome. Okay, so that's important to remember, and that's important to remember because of something we're going to talk about during meiosis that introduces a little randomness into the process, so that you don't look just like your siblings. Somatic cells are diploid. That means they have two complete sets of DNA. That is, they have a complete set from your father and a complete set from your mother. Gametes, on the other hand, are haploid, which means they have one set of DNA, but it's a mix of DNA from your father and your mother. So, that's a major difference to remember. Homologous. Homologous describes two chromosomes that share all the same genes in the same places, but they might have different versions of the genes, okay? Those different versions would be think of it this way. If there's a species of flower and some individuals have white petals and some have purple petals, the gene for petal color will be in the same place in both individuals. It's just one will have a version for white and one will have a version for purple. That's what homologous means. The chromosomes that carry that gene are homologous. Centromere is the spot where in diploid cells, the chromosomes actually attach. So your father's and your mother's DNA have a point where they attach in the middle. And that's what gives the characteristic sort of squiggly X shape that we're used to. The Y chromosome, interestingly, is actually an X chromosome that's just had, at some point in evolution, lost most of the bottom part and just fused together so it looks like a Y. Mitosis, somatic cell division. Meiosis is sex cell or gamete division. Okay. So, here are chromosomes. Okay. Each pair of a chromosome segment here consists of sister chromatids. These are homologous sequences of DNA. They have the same genes in the same spots. Okay. As you can see here, here are all of the human genomes. We number them. It's actually in order of size. So 1 is the largest, then comes 2, 3, 4, and so forth. The spots in the middle here where they seem to sort of match up are the centromeres. And you notice some of them look more like H's than X's, but that's fine. And then you've got down here, you've got the X and the Y. In this case, this is a male individual, so it has an X, one X, and one Y. The Y is the stubby little one. Okay. Now, during mitosis, which is somatic cell division, a cell goes through its life cycle. Most of its life cycle, it's just growing, okay, and doing its stuff, what it's supposed to do. But there comes a point where it gets ready to reproduce itself. And at that point, several things happen. First, this loose chromatin you see inside of step 1 bunches up into individual pairs of chromosomes in step 2. You can see they're coloring them 2 different colors because one would be from one parent and the other would be from the other parent. You'll notice too that these little poles form on the sides of the cell envelope. Now, those chromosomes start to line up along those polar lines that are connecting there, and eventually they will split apart so that you will get one half of the chromosomal pair over here and one half of the chromosomal pair over here. And then in the end, you will have two, the nucleus will reform around each of those, and the cell will begin to divide in the middle. And in the end, you end up with 2 daughter cells with identical sets of DNA. These are identical cells. Assuming there hasn't been any mutation in the meantime, these are identical cells. Okay. That's the basic life cycle of a cell. Now and that's what you see here, right? So you've got a parent chromosome, everything doubles, you get these pairs, and then the pairs split off into the individual cells. Simple enough. Meiosis is a little different. It starts off the same. We've got a bunch of chromosomes, 2 full sets of chromosomes. In this case, we're saying 2n equals 6, so we're saying the individual actually only has 3 pairs of chromosomes. Those double, but instead of forming separate pairs, they form into what's called a tetrad. So both parents' DNA are doubled and then combined together. And during this period, because they're so close together, you can see right here, it's possible, because they're all homologous, that means they're chemically very similar, which means it's easy for them to exchange the ends of their chromosomes with one another. And to give you a picture of that, imagine this. This is the process from a single pair of chromosomes perspective. Let's say blue is the mother, red is the father's DNA, and you've got the mother has genes for capital represents a dominant gene, lowercase represents a recessive gene. We'll talk about that in the next chapter. But for now, just understand there's a dominant A and a dominant C on the blue, on the mother's chromosomes, and a recessive A and a recessive C on the father's. Well, during crossing over, they're going to exchange these little legs. And so now we go from having two identical pairs, right? Two pairs of identical uh, um, chromosomes. So, the blues are identical to each other and the reds are identical to each other, to now having four completely different chromosomes, right? Or chromatids. The first has the original dominant A and dominant C. The next has a dominant A. Notice it didn't get exchanged, but sorry about that. My finger keeps slipping. The recessive G has flipped over to this side. So now it is dominant A, recessive C. And then we've got a recessive A and a dominant C, and a recessive A and a recessive C. So now we've gone from having just two possible combinations of the genes A, C. You're either all recessive or all dominant up here, to now each of these is a different combination of them. This is called crossing over, and it is sometimes also referred to as recombination. And it is an important way of mixing things up. So it's not going to create a new gene, but it will create new combinations of genes that will be novel. Okay. After that, the genes or the cells go through an initial division, so that now they've got a normal distribution of the gene of the chromatids, and then they go through a second division so that the eventual gamete has just one set of chromatids. And you'll notice each of these gametes has a separate, independent set of these chromatids. No two of these gametes are identical. That's an important difference between mitosis and meiosis. Mitosis, the goal is to create a Xerox copy. Okay? Meiosis, the goal is to not create a Xerox copy. The goal is to introduce variation. So, now, this last step sometimes goes wrong, and the individual chromatids here don't always split up correctly. So you can see here, we'll just talk about the long red one, and the largest of the chromosomes we've got here. One side of this, the long red, all red, sorted into this gamete on the far left, and the long red with a slightly blue end sorted into the next gamete. So that's what should happen. Sometimes, though, the long red and the red with a blue tip don't split up, and they both end up, say, in the gamete on the far left, in which case the next gamete will get no copies of that chromosome. And that means you will have a gamete with two copies of one of the chromosomes and a gamete with no copies of one of the chromosomes, which results, when they fertilize with a normal gamete, in either an individual with no copies of a particular chromosome, of one copy of a particular chromosome, or of three copies. One copy is called monosomy, three copies is called trisomy. And an example of each of these, the first is Turner syndrome, where a phenotypically female individual inherits an X chromosome, but doesn't inherit a second sex chromosome. So they only have one sex chromosome. This often prevents the individual from reaching full physical maturity. And there's some other sort of implications as well, but we won't get into those right now. The important thing is that it's the result of having a single copy instead of two copies of the sex chromosomes. Trisomy, an example, is Down syndrome, also known as trisomy 21, because it results from having three copies of chromosome 21. So each of these is the result of this disjunction going wrong. This disjunction, we call it a non-disjunction error or non-disjunctive error, because it doesn't disjunct. Is that a word? Disjunct? It doesn't separate. Okay. So why are we bothering with genetics and all of this? One reason is because we now rely heavily on a lot of molecular tools to understand ourselves in the present and in the past. Key to this was the discovery in the 1980s of the polymerase chain reaction, or PCR. It's a method for amplifying small amounts of DNA. Basically, you can start with just a couple copies of DNA, and by repeatedly heating and cooling that DNA in a mixture of free-floating nucleotides, heat will cause it to unzip, and then with the right enzymes and nucleotides in there, as you cool it, they will form two new double strands. And then you heat it and those two strands split, and then you cool it and you end up with 4, and then 8, and then 16 and 32 and da-da-da-da-da. And within a day you can have 1 million copies of a single copy of DNA. This has been a major advance. It really, because honestly, DNA is incredibly fragile. It is what allows criminologists to sort of solve crimes on biological samples that are decades old. If they've been stored properly enough, you can recover very small amounts of DNA and amplify them till you have enough that you can actually sequence it and come up with the dominant features of that genome. PCR has also had a huge impact on ancient DNA discovery, and I will talk about that in a little bit. It is a way, with certain kinds of fossils, we can remove DNA from them. It's always highly degraded, but using PCR, we can get enough of it together that we can do what's called a shotgun approach and sequence it and look for where the pieces overlap. And that's how we can sort of build up an entire strand from a bunch of tiny pieces. It's like piecing a puzzle together. Okay. So PCR has been major in terms of the Human Genome Project, describing the chimp genome, which occurred about 12-15 years ago, I think. The Neanderthal genome, which was just completed in 2009. This is why I said everything I learned in the early 2000s, completely up in the air. Everything we know about Neanderthals has changed. Everything. A whole bunch of assumptions we made about them in the past turn out to just be wrong. Just be absolutely wrong. And in fact, turns out that something that all of us had ruled out for decades, the fact that Neanderthals and ancient humans might have interbred, turns out to be true. And we have the evidence in the genome. Now, ancient DNA is very difficult to study because oops. Now, the study of ancient DNA started off not by taking nuclear DNA, but looking at mitochondrial DNA. And the reason is, in every cell, every somatic cell, you just have 2 copies of nuclear DNA. But you have potentially hundreds or even thousands of copies of mitochondrial DNA in each cell. So you have a significantly better chance of some of the mitochondrial DNA having survived. Plus, mitochondrial DNA has some very highly variable regions which allow us to do things like measure for molecular clocks, and even more importantly, sort of, look at, if you're looking at relatively small timescales like 100,000 years, I know that sounds ridiculous, but in evolutionary terms, that's a relatively short amount of time. But if you look at places that are more likely to mutate in a genome, you're more likely to sort of see differences on that time scale than you are in other places. So it's a better place to look to do your analysis. And mitochondrial DNA has a lot of those. Lastly, for other studies, mitochondrial DNA is especially good because it is passed on clonally. It's highly preserved from mother to child in every generation. Only women can pass it on because sperm only contribute genetic material. They do not contribute their organelles. The organelles belong to the ovum, which is produced in the mother's body. And so males cannot pass on their mitochondrial DNA. Only females can. Okay. Now, why is that important? PCR is essential to this because DNA is extremely fragile. Water can damage it. Heat can damage it. Alcohol, ultraviolet light, all sorts of things can cause it to erode and degrade. And it just degrades on its own, even in the absence of all those things. So it has a natural decay rate. The downside of using DNA, well, the upside of using DNA is that it's a highly reactive molecule. So you can do all these neat sort of things like transcription and translation, right? But the problem for looking at ancient DNA is that means, it also eventually breaks. The other problem, if you're looking at things that are really closely related to humans, but humans are collecting the DNA from those, from fossil humans, contamination becomes a huge risk, because how do you tell whether you're looking at genes that really were shared between this hominin and us, or whether you're just looking at, Jack the curator's hair follicle that fell into the bath, right? So that becomes a huge concern. But PCR has helped us with so much of that. That said, the real importance and the real reason we want to look at genetics is because genetics and DNA are the sort of engines of evolution. It is actually the very stuff evolution is working on ultimately. That is how we get new traits. It is how new traits are passed on, and it is how natural selection operates. It is operating directly on the results of the interaction between genes and the environment. So, that is why we start with this sort of molecular biological sort of stage here. Now, in the next chapter, we're going to go one step up, and we're going to still look at genetics, but we're going to look at the sort of individual level and look at the transmission of traits from one generation to the next. What are the patterns? How do we study that? What are mutations? Let's get into mutations a little more. What are they? What are the implications of mutations? What are the different ways they can manifest themselves in the genome? And that'll set the stage for the subsequent chapters where we're going to discuss population genetics. So again, going up from the individual to the population, and eventually all the forces of evolution. And eventually, finally, we're going to talk about how evolution and natural selection are forces at work in humans today. Okay. Hope you're looking forward to it. I know I am. Take care of yourself and have a great day.